Okay, we're carrying on with that paper, question 5. You've got red blood cells. So it's the June 23 paper 1B for biology. We're looking at question 5. Red and white blood cells are two components of human blood. What are the two components? Are there? There's plasma, the liquid part, and you have platelets. You can have other things, to be honest with you. Okay, so let's carry on. You've got some white blood cells that destroy pathogens. What did we say a pathogen was? Can you remember? It's a disease causing organism. Right, you've got to know that. Right, we've got two types of white blood cell. So remember, white blood cell. You've got two types. You've got a lymphocyte, and a phagocyte. Lymphocytes make antibodies and turn into memory cells. Phagocytes engulf pathogens and then they kill them, digest or kill them, dispose of them. Right, so we need to describe what's going on. So if you look here, the phagocyte, that's that cell there. Have you noticed these important things that you need to know? You've got a lobed, if I can spell it, nucleus. It's not your normal round one that we draw like this. And you've got digestive enzymes. I think those are digestive enzymes, I think. It's not clear. So what happens is it engulfs the pathogen, or in this case is a bacterium. So bacteria is plural, bacteria is plural, many. I'm just learning this myself, so I was just reinforcing it now. Then you've got these digestive enzymes will attack this and break it down, and then it expels the waste products of the digestion process. So we can say in stage 1 or stages 1 and 2, the phagocyte engulfs the bacterium, right, and then in stage 3, digestive enzymes are released to break down the bacterium. And then finally in stage 4, you get the waste products of the digestion process are expelled or are you can't use the word excreted. Well, no, they're not excreted. Egested? Oh, they've just in the mark scheme they put expelled, so we'll stick to that. Right, okay. Oh, right. Here we have a picture of a red blood cell. A red blood cell. Right, okay, let's have a look. Right, the actual diameter. Oh, okay. The actual diameter of this is 8.1 micrometers. Right, this is the British spelling. Micro, they use this Greek symbol mu or mu. We call it mu, but I watched a video where a, a, a person from Greece, a Greek person, said it's not a cow, it's not mu, it's me. I hope I've not butchered that. So it's 8.1 times 10 to the minus 6, or 8.1 divided by 1, 1, 2, 3, 4, 5, 6 zeros. That's what that minus means. This is where you've got to be comfortable with your maths. Okay? Or you can write no point, no, 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 no. How many notes? Right, let's have a look. 3, 4, 5, 1. There you go. That's how many meters there. Right, we've got that. Right, so magnification. So we'll just write here. Magnification is what is it called? Image size. I've not come across questions like this before, so in IGCSE. So the first time I'm coming across, oh sorry, I lied, second time. Image size and actual size. So I think it would be good to know this by the looks of it. Although this year they're giving so they've measured it at 5 centimeters. What do you call it? Image size. They measured it at 5 and if we turn it into meters, it becomes 0.01257 meters. Right, the actual size is here, and that is there. So what we need to do is write down, so let's write this down. Actual size equals 5.7 centimeters, which is 0.057 meters. That's step one. Image size, let's convert it to 8.1 micrometers or micrometers, and then you have a not 81. I'm gonna have to check this. Meters, and then the actual calculation is 5.7 divided by I'm gonna just be a bit cheeky and use that number instead. Right, let's run with that. And let's try it, so just bear with me. Right, I forgot to change this. I used the wrong number here, so I'm gonna, right, yeah, what I'll do is I'll be cheeky again. I'm gonna write that in meters, and the answer is 7.037 recurring. So you can write it as 7037, more than enough. That's the magnification. Right, I've seen another question like this recently. So this I've not come across before. I saw one 2 days ago, and to be honest with you, I think it was some sort of bacterium, if I can remember. I hope I'm not mixing it with something else. Oh yeah, I did that with the student. I remembered. Okay, let's move on. Stop wasting time. Right, patients' reduced number of cells are classed as anemic. There are 4 groups of patients. So there's an investigation going on to measure the percentage of oxygen saturation of the blood and the rate of tissue deoxygenation. Right, what they're trying to say is the rate of tissue deoxygenation is the measure of how fast oxygen is lost from tissues. There's 4 groups of patients. Non-anemic, normal. Anemic, normal. Non-anemic, slow. Anemic, slow. And here are the results. I think it's going to be one of those where I might have to do this, I'm afraid. See if we can squeeze that in somewhere. Right. Can I put it here? Yes, I can. Can I reduce it? Not really. Not a very good idea. Right, if I can reduce it. Yes, we can. Can we? Yes, we can. Not reducing it too much. So we're being asked to discuss the relationship between anemia, blood flow, percentage of oxygen, blood saturation, rate of oxygen tissue deoxygenation. You should refer to, and how many marks? 5 marks. Right. So patients who've got normal blood flow, non-anemic, anemic. You've got 81% versus, so normal blood flow generally, so right, this is what they're trying to say to you, right? So normal, oh, word. So we need to answer this in a way that we can pick up maximum number of marks. Normal blood flow is higher overall, if you add them two, than slow blood flow. So we can write that down. So normal blood flow, percentage oxygen saturation is greater, is higher than or when compared to slow blood flow. So do you know, if you're in these questions and you're thinking, what can I say? Just do some logical waffling, because teenagers are superb wafflers. They can get out of any detention and trouble. So right, normal blood flow rate of oxygen or deoxygenated is higher in non-anemic patients. So non-anemic patients, the saturation is higher. So that means anemic patients lose oxygen quicker at a quicker rate. So we can write this down, yeah? Normal. So remember, we started comparing the 2 different groups. Now we're comparing within the group. Normal blood flow, normal blood flow rate of deoxygenated is higher in non-anemic patients, is higher in look, can you see this is higher non-anemic patients. Right, what else can we spot? Let's have a look. Percentage oxygen saturation lower, percentage in normal blood. Percentage oxygen saturation lower in anemic blood in normal and slow blood. Right, okay. So what they're saying is this, and it is, right, let's get rid of this now. So just look at this now for a second. What now they're doing is they're comparing normal and slow blood, anemic and anemic. So they're saying, let's get another color, let's get another color there. Let's get another color, let's get another color. This color's better, it stands out a bit more. Right, okay. So we're saying normal, so we're comparing that normal blood flow anemic patients, normal blood flow anemic patients have a higher blood saturation level, have a higher percentage blood oxygen, I'm writing those all in the wrong order, saturation compared to slow blood anemic, compared to slow blood anemic patients. Right, okay. Right, so now they're saying, so now what they're doing is they want to compare what's the difference between anemic and non-anemic in normal blood flow and anemic and non-anemic in slow blood flow. So now we can say there is a greater difference between non-anemic, so non-anemic and anemic. Anemic patients in normal blood flow compared to, you can put figures in if you want, anemic and non-anemic and anemic patients in slow blood flow. Okay, so that's the fourth point we made. Mean rate of tissue deoxygenation per minute, number of patients. What else does it say? So what that will mean percentage, mean rate of tissue deoxygenation per minute. Deoxygenation saturation. So what? Right. Why would that be? And so now the next part is we can look at, so that means right, let's go back and have a look. Does it even say did no, it doesn't. I thought it might. So right, okay, we're going to carry on here. So according to the math scheme, they start discussing there are fewer, and it's normally linked to, they say, deficiency in iron. And remember the hemoglobin or hemoglobin molecule is made, contains iron. That's what gives it its red color. So if you've got deficiency in iron, you'll have less hemoglobin or even red blood cells. So there are fewer red blood cells in anemic patients. Right. So there's less capacity to carry oxygen. Also, anemic patients anemic patients tissues deoxygenate faster. Right. Right, okay. What also could, they've also put in, but we don't know that they've not presented it. That's what's concerning me. And use your biological knowledge. Okay, right, fair enough. So we do know things like lifestyle choices, they can lead to sort of health problems. Lifestyle choices, such as smoking, your gender, your diet, overall health can affect the blood's ability to carry oxygen. Okay, that's cool. That's it. That is it. It's all there. It's I've not seen a question like this before, so it's worth looking at. Right, 23 minutes. Yeah, let's see if we can get this one in. You've got a pea plant. Flower position is a genetic feature. So here, the flowers are axial, they're around there. Here, the flowers are at the end, terminal. So they've said A and T. In a first cross, scientists crossed pea plants with axial and terminal. The first cross produced 1,120 offspring. All of these offspring plants had axial flowers. Right, does it say? No, it doesn't. Okay, so what we do is we draw some sort of table like this. Right, we can, right, there's two ways of doing this. We can say the parents' genes or gametes, right? So axial and terminal. Axial we'll call A, and terminal we'll call A. Right, remember all these were axial. Right, what are the right, let me reword that. Parents' genomes or genes, genomes. Right, and then we can look at the parent genes that they're contributing. They're going to contribute a dominant and so axial and then non-dominant terminal. So now we need to look at what the kids are going to be, the offspring genotypes or genomes. Genotype. Again, let me just genotype. Okay, so we've got this. And they were all tall. So they're gonna I think they're gonna have AA. So what we go, if we can go to the something called a Punnett square. No, there's no E. I always get the spelling wrong. Come on. Gotta wake up one day. Start spelling properly. Square. So we can call this parent one, which was axial. Axial is capital A, two genes, and this one is terminal. So you've got little a and little a. So now if you cross this, an A and an A gives an A and an A gives an A and an A gives an A and an A gives. So you've got 4 plants that have got that are heterozygote. Let me use that word, heterozygote. That means there's one of each, and the other one is homozygote. Homo just means same. Hetero means, I don't know, hetero not same or different. Okay, that's not a problem. And that's it, you're going to get AA. So that's why all the, look here, all the offspring, the kids were axial. So that means that that is the dominant one. So you can do it like this. You can do a Punnett square, which will give you the marks. Right. Oh, right. Now we've got scientists allowed the offspring from the first cross to self-fertilize. So for example, there's a plant. So what you do is you take the pollen from this flower and put it onto that flower. And you take the pollen from there with the brush. And you don't allow any mixing, so you could put a bag over them or put them in a separate little mini sort of like container or glass house. Right, this second cross produced and 608 were axial. So what they're saying is 858 minus 608 gives you 250 terminal plants, while 608 were axial. So they want the ratio of axial to terminal, axial to terminal, axial to terminal. and they want in the ratio of 1. Because they want that 1, we're going to divide this by itself, and we divide this by 250 as well. So we end up with 2.43. There you go. Explain why the ratio of plants with axial plants with terminal plants is different than the expected ratio. Right, so 3 marker. Right, again, I've just looking at the mark scheme, I've not seen this before, so what we want to do is work out the ratio. The expected ratio is 3 to 1. Right, come over here and have a look. So that's one parent and this is another parent. Just say that's pollen and that's the flower. So if you take A and A, you get AA, that is axial, AA, axial, AA, axial. Why? Because you've got the capital A, that makes it an axial plant, and then you've got 2 small a's, that makes it a terminal plant. So we're expecting axial, 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 and terminal. We expect a ratio of 3 to 1, but we got 2.43 to 1 because fertilization is random. Fertilization is random. So where's the third mark coming from? 1, 2, 3. So we can say maybe because also the same fewer axial flowers or plants survive and then germinate. So they could have that. You could have 2 things. First of all, it's random, and secondly, fewer axial plants might have survived. Maybe they're not as robust. That's a tough one. Right, okay. We've got our classic columns. Right, so we want to discover axial produce more seeds than terminal. So we need to design an investigation. Right, right, first of all, right, you can start off with I will use, which organism are you going to use that you're going to control? So I will control the type of organism I use, which will be an axial plant and a terminal plant. So I will use many axial plants and many not radial terminal plants in this experiment. That's his experiment, by the way. So look, they're saying include details and try to write in full sentences. That's a full sentence. Now, which organism would you use, or what would you keep saying about the organism, or right, about the organism that you've used up here? Look, them and them. I will make sure that I use, and this typically it'll be age or species or mass, yeah. I will use the same species and age of plant or plants. Right, how can you make sure your calculation is reliable? I will use many axial and many not radial terminal plants. So can you see what we've done so far? Right, the organism that you're gonna sort of keep an eye on, the independent variable, I will use many axial and terminal plants. Right, what you're gonna do with that independent variable, you're gonna make sure to make sure it's fair, you've got to use the same species. It's no good having two different species because that could affect it, and age. It's no good having terminal as really young plants and axial as really old plants that are about to die and they can't produce any a good sort of crop. They've gone past the optimum. And this is a standard. This learn this. Let me change color of that. I will use many axial and terminal plants. Right, now this is how you finish it off. Just bear this in mind, okay? Just look at this. I will use many term plants. Remove any anomalies. Anomalies. Someone asked me what's an anomaly. It's away from the other data. So for example, I'm running 100 meters. I'm running it in 10.1 seconds, 10.5 seconds, 9.8 seconds, 10.12 seconds, 10.6 seconds, and then suddenly it takes 15 seconds. So this here is an anomaly. I'll ignore that. Or if I had another one at 5.2 seconds and Usain Bolt's going, what? I'd ignore that. Right? So these are concordant results. Well, okay, let's not use that word. These are results that fit the trend. Okay, so anomaly is something that is away from the other data or from the normal data. It's away from it. So you remove them and then average out results. Right. Now what you're going to do for the M. There's normally I will collect the seeds and you can count them or weigh them. Count them for each plant type, and then you can say this is all continued on one sentence over a period of just say over a growing period of 6 months. So it's no good leaving one for 3 months and another one for 6 months. Right, now what do you keep the same? This is the key now to make it a fair test. I shall ensure the temperature affects growth. Temperature is the same. The humidity, so water levels, humidity, the CO2 levels, just say, okay, the what do you call it, fertilizers or nutrition levels. You can say fertilizer, and just say make sure that they've got the same soil. I shall ensure the same, let's just put it once here, same temperature, humidity, CO2, fertilizer, and same soil type. These all affect growth, so you have to keep them the same for both the plants and all the plants really to make it for a fair test. So look, this is your corms structure. It's gonna come up again and again and again, learn it. So what's the organism you're dealing with, and how are you gonna keep it steady? So this is what your control is, in terms of you're gonna keep the same, it's independent, and then these are your dependent variables, the ones that you're gonna measure, and these are your control variables. But do it in that order and you can't go wrong. You'll pick up most of the marks. Right, okay, we can call it quits at that. So question 5 and 6. Well done.